Hi, folks. Welcome to the mitochondria. The mitochondria we've seen before a little bit in gluconeogenesis, but today we're going to go and we're going to stay for a while in the mitochondria. We're going to go through the pyruvate dehydrogenase complex. We're going to go through electron transport in the Krebs cycle. We're going to go through fatty acid breakdown. So we're going to be here for almost the rest of the term. Mitochondria really are the powerhouse of the cell, if you will. And so we have now gotten out of the cellular cytoplasm, and we'll come back when we do fatty acid synthesis, but we're going to stay in the mitochondria for a little while, and we'll be doing quite a bit of understanding of what's going on in the mitochondria. But to start out, we probably need to understand what a mitochondria is. Mitochondria are effectively these little rod-shaped bacilli, a little bacteria-like creatures that live inside of our cells, and they're symbiotic with us. So they have two membranes. You have the outer mitochondrial membrane, so I'll call that the OMM. That's a membrane that's pretty porous and is relatively passable to lots of different kinds of materials. And then we have this kind of funny looking inner membrane that's all kind of squished in there. If you've ever tried to squish something big into a small space, you have to kind of fold it and squish it in and pack it in there. And so the inner mitochondrial membrane is actually quite large, and it has these folds in them that are called cristae on the inner mitochondrial membrane. That give you more surface area. And the inner mitochondrial membrane is actually very high in protein. It's about 50% protein, or 50 to 75% protein. And the rest of it's being lipids, right? So it's very packed with protein. This is where our electron transport chain happens. Fatty acids, a large part of those happen here, but largely it's electron transport. There's also translocase enzymes. Our ATPases, that's where our ATP is made. Those are all embedded in that membrane. And so that inner mitochondrial membrane is actually very selective for what gets in and out, right? So almost anything can pass the outer mitochondrial membrane. It's just kind of like I don't know, like a coarse net. Large things can't get in and out, but small things pretty much can pass freely. Whereas the inner mitochondrial membrane is the bacterial membrane. If you know anything about mitochondria and the endosymbiosis theory, an important theory is that these mitochondria at some point in the evolution of eukaryotic life, which is life with a nucleus in the cell, essentially engulfed bacteria, but didn't destroy it, and it essentially had the stuff inside the cell for the bacteria to live on. And then the bacteria provided ATP and other kinds of nutrition for the host cell, and so we became symbiotic. We became we started to rely on each other, and so now we can't be separated. And so the actual cool thing about this is that mitochondria are little independent little creatures. They can't live outside of our cells. We can't live without them. So we become mutualistic, right? We feed them, they feed us. Everything goes great, and now we're inseparable, right? We can't live without our mitochondria, and the mitochondria start to die, our cells die. That's what apoptosis really is. So a couple things we need to highlight here as we're going through some terminology. The cytoplasm of the mitochondria is called the matrix. The matrix is the inside part. That's where the Krebs cycle happens. And of course, many other cytosolic or soluble reactions happen in the mitochondrial matrix. And then between the two membranes is what we call the intermembrane space. Or the IMS. We'll talk about that a fair bit. And that's a place where we actually build up a gradient potential. We'll have a lot of protons out there, and then we have fewer protons inside. And we use that proton gradient to do a lot of interesting work inside the cell. And the last thing we need to know is that mitochondria have their own little chromosome. That's called mitochondrial DNA. You might have heard about this if you're like me and you like to watch Forensic Files or any of these kinds of crime shows. Mitochondrial DNA is maternal. The maternal oocyte, or the maternal egg, during the point when you were fertilized, carried the mitochondria, and your father's sperm had none. And so you only get mitochondria from your mother, and they divide on their own. And so they pass down as a maternal lineage from the mother, just like the X chromosome or the Y chromosome passes down from the male line. So the mitochondrial DNA is a good maternal marker because it only comes from the maternal line. Okay? And so keep this in mind as we start to talk about stuff that's going on here. So what I'm going to do is I'm going to zoom in a little bit on the inner membrane here. Make a little blow up, see if I test my artistic skills. Okay, so we have two membranes. And so here's the outer mitochondrial membrane. Here's the inner membrane. Okay. So here's the inner mitochondrial membrane. Here's the intermembrane space between the two. And remember that the outer mitochondrial membrane is pretty porous. So let me draw that with some big openings in it, I guess. Just lots of stuff can just come in and out. It's a bacterial outer membrane, and I guess it's very permissible for things to pass through, okay? And so anything can pass in and out of this, right? So between the host cytoplasm and the intermembrane space, those two are pretty well connected. There's not a lot of selection. Whereas the inner mitochondrial membrane, only things that we allow in and out can pass, right? Because now we're entering a new organism. We're going into the mitochondria, or essentially a bacterium. And so we need transporters that are specific. And so, for example, pyruvate from the cytoplasm came from glycolysis, which is a cytoplastic process and pathway. And so it needs to come in. It'll cross into the intermembrane space. And then we have our pyruvate translocase enzyme. Which brings pyruvate into the mitochondrion. But it does this as a co-transporter with hydrogens. And again, we talked about how we have high hydrogens out here and low hydrogens out here, or in the matrix. And so hydrogens can be used to pump pyruvate into the cell, into the mitochondria, even when there's a lot inside already. And so we can use the proton gradient of the intermembrane space to the matrix to pump pyruvate in, and translocase is going to do that for us. But because of that, that makes it a unidirectional transporter. So we saw earlier when we were talking about glycolysis that we have to trap glucose inside of the cell because we could worry about it leaking back out. But with H+ being pumped into the cell, that keeps this unidirectional. So we pump only in because that's the gradient of protons that are pumping, right? We're not going to pump protons out into a high gradient. That's going to be thermodynamically unfavorable. So instead, we're going to use protons to keep us pumping inwards all the time, okay? And then we're also going to try to keep the pyruvate inside the cell out of the equilibrium, so we don't have to worry about it sneaking back out on us, okay? And so this is where the pyruvate dehydrogenase complex comes in. The pyruvate dehydrogenase complex is a complex made up of 3 different enzymes. One is pyruvate dehydrogenase. What does dehydrogenase use? That's right. You're so smart. Any dehydrogenase is going to use NADs or NADPs or FADs to do the dehydrogenation, the removal of hydrogen. And so the pyruvate dehydrogenase is going to do 2 different things. This is very similar to what we saw with transketolase in the pentose phosphate pathway. Pyruvate dehydrogenase is a TPP enzyme, which means it does alpha cleavages, carbon-carbon bond cleavages. And in this case, it does alpha decarboxylation. So we remove the carboxyl group from pyruvate in this step, but we also use the NAD to NADH. The pyruvate dehydrogenase complex is a catabolic pathway. It's a really short pathway. It's only one enzyme, but it's a feeder pathway between glycolysis and the Krebs cycle, right? And so ultimately what's going to happen here is we're going to be making our pyruvate into acetyl-CoA. And I write this as S-CoA because it's a little, I mean, I think it's the right way to do it. We connect through what's called a thioester linkage onto our acetyl group. And so together we call this acetyl-CoA. And that is the foundation for the Krebs cycle. It's also what we get from to synthesize fats, and it's used in other pathways as well. But ultimately, acetyl-CoA is the common thing that's kind of trapped inside mitochondria and designated. You can think about CoA being kind of like the phosphate in glycolysis that traps glucose inside of the cell. With CoA on it, essentially you need a special transporter to move it, and there is no acetyl-CoA transporter specifically across the inner mitochondrial membrane. You need to switch out CoA for some other carrier to be able to move it across the membrane, okay? So we'll see that as we get deeper into the material. But keep in mind that we have two outputs here. We see a loss of CO2 from pyruvate dehydrogenase. So I'm going to draw my little arrow here. CO2, NADH, our products, and NAD coming in. But we also need to put the CoA on it, okay? And so that's kind of what happens in enzyme number 2. Enzyme number 2 is our lipoamide acetyltransferase. And this is a new class of enzyme with a new cofactor called lipoamide, which looks like this. That's 1, 2, 3, 4, 5, 6. And in this case, this is a full lipoamide, but this can also be reduced to the dihydrolipoamide like this. So this is what we're going to call dihydrolipoamide. And this is an acetyltransferase, which means that essentially what it's going to be doing is taking the acetyl group from enzyme 1, and moving it on to CoA. Okay. So we need lipoamide here, and we're actually going to call this dihydrolipoamide acetyltransferase. And so we're going to need a CoA as well here. So we're going to put that in our kind of shorthand for the pathway. So our pyruvate's coming in, reacting with CoA and NAD to make CO2 and NADH. And at this point, we're going to be not releasing CO2 and NADH like we did in the first one. We're going to be releasing our acetyl-CoA as a product. Okay. So there are 3 different enzymes here in the complex and 3 different products. And each one of them is responsible for a different role. Here we have generated our 3 products. We've generated acetyl-CoA, we generated a CO2 and an NADH. And remember that an NADH is worth about 3 ATP each. We'll see whether that holds up as we get a little deeper. And essentially what happens is, I've kind of skipped the step here. This NADH is not necessarily made here in the dehydrogenase step, okay? We're gonna have to wait for that for a little bit, okay? This does not actually happen here in this complex, okay? Even though it's called a dehydrogenase, and this is kind of why I put that there, you might think that it's a dehydrogenase and it should do NAD to NADH, but this actually happens in enzyme 3, okay? So don't get confused about this. It's a weird nomenclature, I understand that, but keep in mind that what's happening here, that the NAD to NADH stuff is happening in enzyme 3, which is our enzyme for generating NADH. Okay. So, just a second. What we're going to see here is our third enzyme, which is the dehydrogenase. So dihydro or dihydrolipoyl, this is the lipoamide, dehydrogenase. This is the one that actually is going to be doing the NAD to NADH. And it does this through using an FAD cofactor. So we've mentioned this before. FAD is used as essentially a placeholder. It's bound to enzymes most of the time. And so FAD is going to always be there when you need it. NAD is floating around, coming in and out. We can't always depend on it showing up on time. And so we're going to use FAD kind of to hold on to the electrons until NAD can take it. So we're going to do FAD to FADH2. And then we're going to transfer 2 electrons onto NAD to make NADH. Okay. And so the idea here is we could think about this being the recharge, the enzyme, or the recycler. Pyruvate dehydrogenase is going to be the decarboxylase. And number 2, enzyme 2 is going to be making acetyl-CoA. And enzyme 3 essentially is going to be the maintenance guy. It's going to be going back and resetting everything. Okay. So before we get into the actual mechanisms here, I want to kind of talk about what's going to happen. Okay. So the way this works is with our 3 different enzymes, and I'll color code these. Enzyme 1 is our pyruvate dehydrogenase. Okay. We end up starting with our pyruvate. We're going to bind onto TPP. So I'll draw our TPP. We're going to form a acetyl TPP. Okay, so here's, we're going to lose our CO2. Okay. And then we're going to drop that TPP off, and we're going to pick up again. Okay, so pyruvate dehydrogenase kind of does a cycle. Pyruvate binds to TPP and does the decarboxylation to generate, that generates an acetyl TPP, which looks a little bit like this. Now, not necessarily exactly like this. It's hydroxyethyl. You can think about it being the acetyl group. Okay, so here's our TPP. Oops, not that. So OH. And then remember that we have a little bit of a partial negative charge here, and we have our positive charge here. So here's our ethyl group and a hydroxy group, and then we're going to be able to use this to transfer onto a new compound in enzyme 2. Okay. So what we're going to see here is the lipoamide coming around. Okay. So here's our lipoamide. And we're going to be picking up our acetyl group This is an E2, an enzyme 2, our trans acetylase or our acetyltransferase. Okay. Acetylipoamide, and what we're going to do is we're going to be dropping that off. So let me draw this the same way. Okay. CoA is going to come in. And then we have to recharge back to lipoamide, and that's where enzyme 3 is going to come in. So FAD is going to be here. We're going to make FADH2. This is an enzyme 3, or dihydrolipoyl. Dehydrogenase. It's called that because this form is dihydrolipoamide. Okay. FAD, we're going to talk about the structure as we get into the mechanisms here next, but it is essentially a 2 or 1 electron carrier. It's kind of a unique and cool little molecule, which we'll talk more about. But for now, understand what's happening here is that we're doing an oxidation back to lipoamide to recycle enzyme 2 by passing it off onto FAD. And we're going to do that by switching with NAD. So each of the enzymes makes one of the products. Okay, NADH is performed by enzyme 3, acetyl-CoA is made by enzyme 2, and CO2 is made by enzyme 1. Okay, so that's kind of the general theme of pyruvate dehydrogenase complex. There's 3 joint enzymes in the complex. Each one of them does a specific job and generates a specific product for the 3 different ones. So again, let's look at this. We're going to have a balanced equation of pyruvate plus NAD goes to acetyl plus CoA SH. We'll get that. We'll talk about the structure when we get to that, and that goes to acetyl-CoA plus CO2 plus NADH. Okay. Enzyme 1, enzyme 2, enzyme 3. All right, so that's kind of the general overview of what's happening in the pyruvate dehydrogenase complex. As pyruvate's brought into the mitochondria, this happens to it. And again, slapping that CoA onto pyruvate to make acetyl-CoA is how we trap it inside the mitochondrial matrix. And so we want to be sure that, that this happens as quickly as possible. And so that typically is happening at the same time. After pyruvate enters the matrix, pyruvate dehydrogenase complex is able to do its work right away on it. And then after that, we're able to use acetyl-CoA for whatever is needed. Okay. So in the next video, what we're going to do is talk about some of the cofactors and go into the mechanisms of reaction.